Today we're going to cover the information on immunity in chapters 15, 16, and 17. By definition, immunity is the ability of your body to protect itself. Now, when we're looking at immunity, there are two types, and the language I'm going to be giving you is a little bit different than your book. I'll explain how they differ once we see the material. Now, when it comes to immunity, the two types are nonspecific and specific. Now, your book says innate and acquired, and they use innate to mean nonspecific and acquired to mean specific. But we're going to see that innate and acquired are really, when you really get into the words and what they mean, are forms of specific immunity. So our nonspecific immunity, to me, is a better definition than innate, and you'll see why as we go through it. Now, with nonspecific, it simply means the immune response, no matter what the infectious agent, is exactly the same. This will become more easily understood when we get into some of the examples. Specific means simply that the immune response depends on the infectious agent. How we respond is dependent on which organism or which type of organism is making you sick. Now, when it comes to specific immunity, we are going to note that there are two types, and the two types are innate and acquired. So again, the language your book uses, we're going to use, but not quite in the same way. Nonspecific immunity, again, means that we're going to see the response being the same no matter what the infectious agent. Primarily, the nonspecific defenses are around the portals of entry. Now, when we look at those portals of entry, we're talking about these things: the eyes, the ear, etc. And so let's talk about all those at once. And what we're going to look at is the defense at each of these portals. Now, here are the portals. Now, when we start off here, excuse me, with the eyes, the big defense are your tears. Now, how do they protect you? Real simple. They flush out whatever comes in. It doesn't matter if it's a virus, a bacterium, or a parasite, tears are going to flush them out of the eyes. Also in the eyes, we see a lysozyme enzyme, which we've already learned over and over, it's the same enzyme found in lysosomes, is a hydrolytic enzyme. So it will cause hydrolysis reactions, which means simply the organic compounds that make up these life forms, because they are all organic, are broken down into monomers. And when that happens, that will destroy the organism. So eyes contain tears. Now, let's go back to innate immunity. Innate means something controlled by your DNA. DNA is the code for protein. Well, tears are not protein. Tears are liquids. They contain salts, and as you see here, sometimes proteins, but that means it's not really controlled by your DNA. It is something you're born with, but it's not really innate that it's coded for in your DNA. It happens as we develop, and we're going to see the same thing applying to the other defenses. So that's why, to me, the better definition is not innate, but nonspecific. It doesn't matter what the infectious agent is. Tears are going to try to flush them out in the same manner. When it comes to your ears, the protection is cerumen or earwax. It doesn't matter what the infectious agent, when it comes in, it gets trapped in the wax and we have to remove it. Now with earwax, we want to remove that by using some type of agent like hydrogen peroxide or even alcohol to soften it, and then a bulb syringe and warm water to rinse it out. What you don't want to do is insert something into the ear canal. As you learned in your anatomy course, and some of you in human biology, the epithelium that makes up the lining of the ear is extremely delicate. And even something like a Q-tip is enough to put lesions into it, and that would make openings allowing organisms to enter into the body. The next opening is your nose, and here we have mucus and hair. Now here, mucus is very sticky. As you inhale, it doesn't matter what you bring in, it gets trapped into the mucus and against your hair. And then occasionally we have to clean it out. Now the question is, how do we do that? Well, most of you think of cleaning it out by blowing your nose, but that's not what the New England Journal of Medicine says we should do. Now, the New England Journal of Medicine says that we should, are you ready? Pick your nose, wait, and eat it. No, I'm not making it up. And for those of you that just got very sick to your stomach, I'm not making light of this. When I heard this on the radio several years ago, I couldn't believe it. And I had to go find the article, and sure enough, it's exactly what it says, but it didn't really say to pick your nose and eat it. What it said was you need to pick somebody else's nose and eat it. Now, the reason is one we're going to learn in chapter 17, and based on the information in chapter 16. The bacteria, viruses, parasites that are trapped in the mucus are very small in numbers. As we learned in the previous chapter, small numbers mean less pathogenicity. So these organisms, although they're pathogens, aren't going to make you sick. So that means we can use them as a means of identification to our immune system. In other words, that booger becomes a vaccine. Now, again, that article is in the New England Journal of Medicine. It was reprinted a couple of years ago, and you can find it online. But it makes perfect sense when you understand how your immune system works. So if those of you who have kids, the next time you see one of your children picking their nose, instead of saying, stop that, say, hey, give it to me and let me eat it. Or better yet, if you've got other kids, have them come in and snack on it too. Moving on, we're going to go to the mouth. Now, the mouth, of course, has saliva. But that is not the defense. Remember what you learned in anatomy. In the mouth, you have salivary amylase. Amylase breaks down starch. Well, starch is not a component of any structure in a bacterial cell, in a eukaryotic parasitic cell, or in a virus. So it will have no effect. So it'll be great on digesting potatoes, but it does nothing against these organisms. So where is the nonspecific defense? Well, it's not in your mouth. Where it is in your stomach. When you swallow, things go into your stomach where they are bathed in acid. Now, acid does not break down food. That's what enzymes do. So in your stomach, you have an enzyme pepsin, which initiates the process of protein digestion. But stomach acid is real good at denaturing proteins, and all of these organisms have proteins. So unless it's an acidophile, when you bathe it in stomach acid, it will be destroyed. The next opening is the urethra. And the urethra, we're going to see urine being the defense. Now with urine, it's simply flushing the pipes. Anything trying to come in through the urinary meatus, the opening to the urethra, will be flushed out. Remember, that opening is just that. It's an opening. It's not a sphincter. It's not open and shut. So things will constantly come in. Now, that's really important because, especially if you're female, it's an issue. Ladies, remember that your urethra is much shorter than that of a male's. So you have to flush your urethra quite often. How often? Well, the study suggests 6 to 9 times a day. Now, that's not an issue if you're sitting at home, but it is a problem when you're working. Because when you're working, sometimes you'll forget to urinate, and holding it is not something we want you to do, because what it means is the organisms can then swim up the urethra and into the bladder, where you'll develop cystitis, or worse, get into the kidneys where you'll develop a kidney infection. Understand, a kidney infection is life-threatening, and it takes a long time to treat. A kidney infection is not a week on antibiotics, it's several months. And the reason for that is your kidney is trying to flush the antibiotics out of your body. It's very difficult to get them from the bloodstream into the kidney to affect the bacteria there. So the best way is to prevent them from getting there by flushing the pipes. The other issue is when you see on the diagram to your right, the anus is real close to the opening where we find the urethra. So what we have to worry about in the case of women is during the process of going to the bathroom is wiping in the wrong direction. Never allow young girls to wipe from back to front, because they'll wipe feces containing bacteria right over that opening. Now, in your fecal matter, primarily what you have is E. coli. E. coli from your gut does not hurt you when it's in the gut. It is actually there to help. But if you put it in the wrong area of the body, such as in the urinary system, it will grow, multiply, and become a very dangerous and life-threatening pathogen. So that's why we've got to keep it out of there. It's also very important on sexual practice. Be very careful the type of sex you practice, because moving bacteria from the anus to the vaginal opening can lead to some very serious urinary tract infections, or even problems inside the vagina, even into the uterus. From there we go into the skin. Now the skin, what makes it a very good defense, and the best defense of your body, is all of the dead squamous cells that are focused there. Remember, they are in layers, stratified squamous, and these cells are dead. Now what that means is if bacteria feed on them, so what? It's not going to hurt you because they're already dead. Viruses will not absorb They can be stuck on the outside, but there's nothing here for them to grow in because these cells are dead. So the point of these cells is to act like shingles on your house. They're overlapped, and so now things can't penetrate and get in, but at the same time as we shed them, everything that's on them goes off of your body. So it's a great means of protection. No matter what is on it, we shed it. And that's why washing your hands works so well. Everything on those dead cells gets rinsed down the sink. From there we go to the anus, and of course the protection is what we just discussed, feces. Anything that tries to come in from outside gets trapped in the fecal matter, and out it goes. So again, just have to understand, in women we have to be very careful about wiping forward. From the vagina, we see that there are acidic secretions produced by Bartholin's glands, and that's why the vagina has a very acidic pH. Now, the problem is if that pH would rise. Now, that can happen, ladies, if you do not practice proper hygiene. And what will develop is what we call an opportunistic infection that you read about in chapter 14. Opportunists are in very small numbers, but when the acids is removed, that allows the proper environment for these to accumulate, and now they'll start growing, and when they do, they're going to produce an infection. And you'll know when you have a yeast infection because it smells like bread cooking in the oven. It has that yeasty smell, so keep that in mind. From there we go to the nipples. Now the nipples are not always portals of entry. They're only portals, they're only open when women are lactating, and so here the defense is milk. Here again, milk is going to flush the pipes and rinse these organisms out. But that's also why when you breastfeed, ladies, you need to make sure you don't use the first milk because it can often contain bacteria. No different than when you do a urinalysis, we always do a midstream catch. You don't take the first urine, you urinate and then catch it so that way we guarantee there are no bacteria in the urine unless you have an infection. If the skin is broken, now it becomes a portal of entry. And when it does, what protects you is blood. When you bleed, blood flushes it out. And that's why you're told when you get a cut, let it bleed for a little bit. Now, folks, we're not talking about cutting an artery. We're talking about just a superficial cut. So keep that in mind. Now we're going to look at some other non-specific defenses. There are three we're going to focus on. Okay, the first is the mucociliary escalator. Now, this defense is in your trachea. As you know from your anatomy and physiology class, the trachea is lined with pseudostratified ciliated columnar epithelium. Now the columnar cells are kind of rectangular and they have cilia on top. Embedded between those are goblet cells that secrete mucus. Now that's very important because this mucus is going to collect on the surface of the tissue. When you inhale, just like in your nose, any particulates are caught inside the mucus. At that point, the cilia is going to beat back and forth and propel these particles up to your throat, where about 200 times a day, without thinking, you swallow. Now, sometimes you might spit it out when you catch yourself doing it, but most of the time you swallow. And when you do, everything in that mucus goes straight to your stomach acid. Now, that's why I always make the argument that the stomach is more of an immune organ rather than a digestive organ. As far as digestion, the only thing the stomach does through constriction is some physical digestion of food, and then it initiates protein digestion with pepsin. But mostly what it does is bathe things in stomach acid to protect you. The second means, and you can see a picture here of this as it actually looks. The second nonspecific defense we're going to focus on is called the inflammatory response. Now this can occur not just on your skin but throughout your body. And as we talked about the other day, this is becoming a real issue when it occurs during the coronavirus. Now in the inflammatory response, we're going to see your nonspecific defenses kick in. And again, with all these, it doesn't matter the infectious agent. Now again, I hope you understand why this is not always innate, because we're not always seeing that this is controlled by protein. When we look at mucus, yeah, it has some protein in it, but it also has a lot of fluids and things. It's not controlled by your DNA. Same thing here with the inflammatory response. What we see is cells responding because cells are differentiated during the process of mitosis, and when they differentiate, they take on these roles, and not everyone's is the same. Now, when we look at the inflammatory response, there's going to be three big things that occur. First of all, it's going to turn red, and that's because we're increasing blood flow, vasodilation. And when blood is brought in, it's going to contain more red cells than white. Now, the red cells are not part of the defense system. but the whites have to be brought in, and we can't differentiate between the two when circulation is occurring. So you're going to have to bring in both. Now, the red cells aren't really used at this point, but because there's going to be an abundance of those, you'll see redness. We'll also see, because of that, swelling, edema. And when it swells up, that tissue becomes a little bit puffy, and this can occur internally too, and you won't always see the edema internal, but it will swell as well. And then, of course, the third thing and the big thing is it's going to get hot. It may be hot to touch, and it will be hot inside. Now, fever is the key, because what fever is your body's means of protecting you by elevating above optimum. Remember, these organisms are all mesophiles, and when we raise the temperature up, the idea is to denature protein, because denaturing protein will kill them by denaturing both the structural and regulator proteins. So enzymes will no longer be able to cause metabolism to occur, and then the proteins that make up the cell membrane, with those being denatured, the membrane comes apart and we lose organization in the cell. So this is a great response. The problem is, when you raise your fever, if it doesn't kill, your body keeps increasing the temperature. And once we get to 104, 105, now that temperature can start to affect us. And that's why we've got to be very careful how we control fever. At the same point, how many of you take something to reduce fever as soon as you get one? Do you understand what we're saying with this? If you get a fever of 102, 103, should you even treat it? Well, honestly, the answer is no. Let it go, because now it will help you more than it will hurt. The last nonspecific defense is where we see interferon being used. Now, interferon is only used against viruses, so this gets a little bit more specific than all the other defenses, but it doesn't go after any one type of virus, it goes after everything. Now, this one is truly innate, because interferon is a protein, but when we look at this again, it doesn't go after any one thing, it goes after all. Now, one of the things we've always noted in history is that no matter what the viral infection, someone survives it, and the reason is because of interferon. Even with AIDS, we had a patient not too long ago that was one who could not get infected by HIV, and the reason for that is the interferon that person produced would not allow the virus to affect them. Now, the way interferon works is real simple. It will either bind to the protein and prevent it from absorption. It will actually destroy the cell that it infects, okay? Or it can also bring in macrophages. Now, macrophage activation, you see, was one of the little bullet points on the inflammatory response. However, I haven't talked about macrophages yet because I wanted to wait till we got to interferon. Macrophages are really unique cells. They are monocytes that differentiate into phagocytic cells. They eat things that are foreign. The problem is macrophages are stupid, and we want them to be stupid. We don't want them just eating anything. So a macrophage will phagocytize things that are considered foreign, but only after the immune system tells them what to eat. Interferon does that. When interferon is attached to the virus, it tells the macrophage to eat this, and it will. When we look at interferon, I want to understand one more thing about it before we leave this. Every person, because the DNA in each of us is different, will produce a different type of interferon. Some work better than others. That's why some people get certain diseases, certain viral diseases, and others don't. So it can be a very good protective protein. And that's why, in regards to the person that can't get AIDS, there are several companies that were vying for his blood because they wanted to get this blood and use it as a means of developing medications to prevent HIV infection. The next thing we're going to look at are the different types of specific immunity. Okay, so when we look at immunity and specific immunity, we're going to focus on two types, those being what we just described as innate and acquired. And when it's innate, you are truly born with it. It's in your DNA. Now, the best example I can give you of this with innate immunity is to think of your pets. And one of the diseases that dogs get is distemper. It's a viral infection. Now, that virus can only affect your dog because the dog has the receptors on the surface that specific virus can bind to. We don't have those. We do not contain the DNA to produce those proteins. So there is no way for the virus to absorb to us. So we have innate immunity. Now, when we look, excuse me, at another example would be, for those of you who have cats, feline leukemia. In feline leukemia, this is a viral infection that affects only cats, causing leukemia. The way the virus binds is by absorbing the proteins on their surface. We don't have those proteins because we're not a cat. So once again, we have this innate inborn immunity. Now, acquired immunity is something you gain in your lifetime. Now, out of these two, which of these is going to be most important? Innate or acquired? Well, that's one of the questions I want you to think about, and it's one we're going to answer when we meet on Thursday of next week. Now, when we get to acquired immunity, there are two types, active and passive. Now, active is the one in which your immune system is going to be involved. Now, we're going to talk later about why this is beneficial, but for both of these, there are benefits and drawbacks. Now, the drawback to active, it's not fast. It's going to take time for this to be acquired. It won't be immediate. Now, passive, we're going to be using the immune system of another organism. It could be another human. It could be another animal. The one thing that's really unique about our immune system, and it's something that I learned when I was teaching zoology, it doesn't matter which mammal you look at. All of the immune systems of every mammal are exactly alike. So whether we're looking at an elephant, a human, or a mouse and a human, our immune systems are identical. And because of that, we can use it as a means of protection. Now, when it comes to passive, the big benefit is it is fast. It's immediate. So that's the great thing. But the problem is, because it's not our immune system, it's going to be short-lived. We won't have it being long-term. When something is active and our own immune system is involved, it gives us a much longer immunity. And we'll talk about how long later on. Now, when it comes to each of these, active and passive, they can be acquired either artificially or naturally. And naturally acquired active immunity, that means you've got the disease and you've undergone the 6 stages. And it's key that you do all 6 because that means you've gotten over the disease. The way you get over the disease is that at the acme, your immune system that we're discussing right here has won the fight. And now you go into decline and then into convalescence. And at that point, you now gain memory to this organism. And that's why you can't get the same disease caused by the same infectious agent. The problem is there are many forms of those infectious agents. Remember that we are all, as humans, Homo sapiens. But there are 7 billion different ones of us on this planet. We're not all exactly the same. Very few are identical twins. So keep that in mind, and the same thing applies to these microorganisms. When it comes to the rhinovirus, which is the cause of a common cold, there are over 200 different types of rhinoviruses. So for you to never get a cold again, you'd have to have over 200. But each time you get a cold, you get permanent immunity, permanent memory to it. That's the reason why we want to have our own system being activated. The problem is it takes at least a week for this to occur. Think about a cold. The most minor of our illnesses takes you at least a week to get over it. Now that's where artificially acquired active immunity comes in. Because if we do immunization, now we can get through the process of our immune system responding to the infectious agent, and it will do this, and it'll still take a week, but it will be a time where we won't be sick. The whole point of using immunizations is to identify these agents to our immune system to gain memory. That way you won't get the disease, but you'll have the protection. Now, if it's naturally acquired passive immunity, that's what we see going from mother to offspring, and that happens through breast milk. So that's why the first breastfeeding is so important. Colostrum contains all the antibodies mom has made in her lifetime. So ladies, how good your breast milk is going to depend on how many diseases you've had and how many that you produced antibodies to. All these antibodies will then be made by you, put into the breast milk, and that will be in the initial breastfeed. Now, that's why that first breast milk isn't white. It's kind of clear because it's nothing but serum. Serum is the part of the blood where we find the antibodies, the immunoglobulins. And now when these are ingested by that child, it gives it about a 4-month supply of antibodies to protect it. Now, the other type of passive immunity is artificially acquired, and that's when we're looking at immunizations, use the right word, immunizations with things like gamma globulin, and we'll talk more about that in chapter 17. Now we're going to look at the properties of immunity. Now there are 4. The first property is recognition. In recognition, this is the ability of your body to recognize self from non-self. So we have to differentiate between the two. Now, the way we do this is to use B cells and the surface receptors on the cells that are coming into your body. Remember, every life form has its own unique receptors. So our B cells can tell what's us and what's not us by looking at these specific receptors. The second property is specificity. Now, this is where we're going to see the immune response. Now we're going to see in specificity our body responding by producing immunoglobulins, or antibodies. Now, the actual term is immunoglobulin, and we abbreviate that capital I, little g. And our body is going to produce one specific immunoglobulin to one specific antigen. Now, what is an antigen? It's simply anything that's not you. Anything. So it could be an infectious agent. It could also be something from another human. This is where we have a problem with blood donation. If you have type A blood, and you're given type B blood in a transfusion, your body's immune system looks at those proteins and recognizes it as being foreign. And it will now respond to it by producing this one specific antibody to that antigen. The next property is called heterogeneity. Now this one's kind of confusing, but when I show you a diagram on a future slide, I think it'll become a little more apparent. Now the problem is there's not just a single antigen. There are billions upon billions of different antigens, and we have to have the ability to identify all of those, recognize all of those, and produce specific antibodies to each one. That's what heterogeneity is. We're going to produce different specific antibodies to different specific antigens. And once we do all this, we're going to gain memory. Our immune system is going to remember, and it's going to be permanent. Now, the reason it's permanent is because the cells involved are white blood cells, and they contain a nucleus. And that means they can divide. And when they divide, they pass on the memory. Now we're going to start looking at the humoral immune system. Now, the humoral immune system is comprised of B cells and immunoglobulins, IGs. Now, when we look at an immunoglobulin, this is what it looks like. It's kind of a Y-shaped quaternary protein. And we're going to talk about the structure that you see here. Now, I want you to note here on this diagram, you see kind of a purple area, you see a light blue area, and then you see what's called the FC fragment on the bottom right. Now, we're going to look at all these structures now. The first structure of the immunoglobulin is what we refer to as the constant region. That's the purple region you see right in the central area, and you can see it labeled. The constant region is going to determine the class that this particular antibody belongs to. And we'll see that on the next slide. In that class, every member will be the same in that constant region. Now, I want you to notice that middle region. When you look at this, you're going to see that these proteins, there are 4 proteins here. Although it's quaternary, not quaternary, it happens to be made up of 4 tertiary proteins, and they are held together by disulfide bonds. And that's what each of those yellow lines with the 2 little bumps indicates. Each of those bumps is a sulfur. So there are 2 sulfurs forming this bond, and that's what holds all of these proteins together. And the constant region will be the same for every member in that class. The variable region is the light blue region that you see on the ends. Now, for each of these immunoglobulins, there will be 2 variable regions, and what they do is bind to the antigen. Now, this is where we're going to see a difference between what we've talked about in the past with proteins like enzymes and antibodies. Remember when we talked about enzymes, I said they are not locks and keys. We only have to have a substrate that has a shape similar to the active site for it to come into that active site. That is not the case for your immune system. Your immune system is a lock and key. We have to have very specific shapes. So the shape you see here of this variable region is not the shape. The shape it will be the exact shape of the antigen it is made to bind to. And I don't mean similar, folks. I mean exact. And that is also going to be a problem for us later on, because when we get things made artificially or even naturally that do not have the right shape, there is no way for them to bind. And antibodies cannot function unless they bind. And it's the variable region that binds to the antigen. The last region is called the FC region, and here we're going to see this is the area where complement binds. Now, complement, note the spelling here, it's not, oh, your hair looks nice today. That's a different type of complement. Here, complement is another immune protein that we'll be discussing when we talk about the functions of antibodies. Now we're going to look at the different classes of antibodies, okay? And there are 4 of those. The first is class IgA. Now, when you look here, you'll notice the middle purple region, that's the constant region, and the shape that it has. Every member of IgA will look just like this. Now, when we look at IgA, it's found on mucous membranes and in tears and in breast milk. So, when we look at these antibodies, these are around the portals of entry. So just like we would see in basic warfare, our body defends itself around the ports. We always want to make sure our defenses in warfare around the areas where the enemy can attack. Well, that's the same thing here in our body. We're going to concentrate our forces at those sites. So we have both non-specific defenses, tears rinse things out, but now we have some antibodies in those tears to help protect you as well. From there we go to class class D, and in class IgD, these antibodies are found on the surface of B cells. Now those, we'll get to the function of those in just a moment when we talk about how this immune system works. Now we have class IgE. Now these are found on the surface of mast cells and basophils. Now many of you may not know what a mast cell is, but a basophil you've heard of. In your anatomy and physiology course, you learn that basophils are involved in allergic reactions. So when we get into chapter 18 and we talk about allergic reactions, hypersensitivities, we'll come back and discuss IgE antibodies. The next class is IgG. Class IgG is found in the bloodstream. So these are going to be the most numerous that we have in the body. And then the last class is class IgM. Now, these are the largest in size, and they are found in tissue. And as you can see, they have, instead of 2 variable regions, there are 10. And that's going to become very important when we talk about how these function. Now, I know you're looking here on the screen and you say, well, you said there were 4 classes of antibodies, but there are really 5. Well, that's true, but one of the classes, IgD, is something you will never make once you're born. And I'll explain that as we go. So the other 4 classes are the ones we focus on. Now we're going to get into the humoral response. Now the humoral response, which again involves B cells and antibodies, there are going to be 2 responses, the first being the primary humoral response. During the primary humoral response, we're going to see many steps. The first step is called sensitization. In sensitization, the B cell is going to bind to the antigen. Now, to understand, B cells cannot bind to antigens. The only thing that can are antibodies, immunoglobulins. So the way the B cell is able to bind is by using the IgD antibody on its surface. When you look over here at the diagram, you will see a whole bunch of different B cells. Each one has a different IgD antibody. Now that is controlled by the DNA of the cell. And you'll notice that each one is slightly different. That is getting into heterogeneity. Your body, when it goes through the process of making B cells, produces a variety of different B cells, each one having a different IgD antibody on its surface. When an antigen comes into the body, hopefully one of these will bind to it, because that means you have the ability to recognize that as being foreign. And if your body recognizes it as foreign, you'll undergo the primary humoral response. If you cannot recognize it as foreign, your body will not respond to it. And now this organism will make you sick and possibly even kill you. So that's why it's so important that our immune system is properly prepared, and making sure we eat things correctly, get plenty of rest, plenty of relaxation, even doing things like meditation can help improve the functioning of this system. Now, once we undergo, with sensitization again, the B cells bind to the antigen using the IgD on its surface. Once that specific B cell is sensitized, now it undergoes cloning. Now, cloning is where the B cell divides and it's going to do so by mitosis. Now that's what we see over here. Now the problem is mitosis is slow. It takes about 24 hours for a cell to divide by this process. So we're going to see it's going to take several days to get enough B cells to respond. Now, unlike what many of you think, we don't need billions of B cells to respond to a disease. What we need is about 8 to 10. Now, to get that many though, one cell going into 2 is going to take 24 hours. 2 cells going into 4 is going to take 48 hours. 4 cells going into 8 is going to take 72 hours. So it's going to take about 3 days to get enough cells to respond. That's a huge problem because bacteria in you are dividing every 20 minutes. If it's going to take this long to respond, those bacteria are going to overwhelm the system before it even has a chance to start, and you're going to die of that disease. So we have got to speed up this process. How do we do that? By using the key cell to your immune system. That cell is called the T4 helper cell, also known as a CD4 cell. This cell is a T cell. T cells do not produce antibodies, but they produce protein, and the protein produced is a hormone called lymphokine. Now your book calls it cytokine. Cyto means cell, but the specific cell it's going to affect are lymphocytes. That's why we refer to it as lymphokine. Now what this hormone does is speed up the division. So instead of dividing once a day, these B cells can divide as many as 3 times in one day. That's a huge change in the speed of reproduction, and it allows your B cell reproduction to catch up to the speed necessary to fight off the infection. Once these cells are cloned, now they undergo differentiation. They change. They're going to change from B cells and become either plasma or memory cells. Now the plasma cells are going to be what produce and release antibodies. The memory cells will not function at this time, but they will be used in the secondary immune response, secondary humoral response, which we'll get to in a moment. Now, once the memory cells are produced, now they're going to produce and release antibodies. Understand that B cells cannot do that. A B cell cannot produce an antibody. It has to differentiate first to do so, so it can produce the specific thing necessary. Now, once these antibodies are produced and released, now the last step, they're going to function. How do they function? Well, in several different methods that we'll talk about on the next slide. There are several ways that antibodies can function. The first and probably most important of all functions is what we call opsonization. Opsonization is when the antibody binds to the antigen, indicating it is being foreign for the phagocytic cells, talking about the macrophages and the neutrophils. Once they recognize it as being foreign, now these cells can act upon it and remove it by phagocytosis. Remember what I said earlier, phagocytic cells cannot recognize things as being foreign, and we don't want them to recognize things. We want them to be told, because we don't want them to do autoimmunity. So to keep them from attacking healthy cells, they will only go after things that are labeled as foreign, and that's what the antibodies are doing. They're like a red flag waving, saying, this is foreign, ingest it. The second means is neutralization of toxins. Opsonization works fine when the organism is present in diseases caused by direct action. But when toxins are produced, now the antibodies have to function in a different manner. Now, toxins are much, much smaller than antibodies. As you can see in the previous picture, an antibody is so small, there's no way it can physically harm the cell by itself. But neutralizing toxins is different because toxins, remember, are proteins or lipopolysaccharides. They are molecules. They are much, much smaller. And what's going to happen here is simple. The toxin, when it's released, is going to be bound up by the antibody, which prevents it from binding to the target tissue. And if it can't bind to the target tissue, it can't affect it. Some other ways that it can be affected is to actually see the antibody cover it completely before it gets there, and it will be removed by way of the urinary system. So any of these proteins that are bound up are taken to your kidneys, where they're put into the urine and flushed out of the body. But the whole point is to keep the toxin from touching the target tissue. Now, when it comes to viruses, antibodies will do something similar, but we now call it the coating of viruses. Viruses, remember, are very small, not much bigger, if they're bigger at all, than the molecules that make up the toxins. And so now again, what's going to happen is that the virus is going to be covered with these antibodies, preventing adsorption. Without adsorption, there's no lytic cycle. Without the lytic cycle, the viruses cannot produce disease. Now, once they're coated, they can be removed in one of two ways. One is to do the urinary system, like we talked about earlier with the neutralization of toxins. The second would be done, as you can see here, by the removal by a macrophage. Once these are covered, the macrophages will remove them by phagocytosis, or it may also be the neutrophils that will do it. The fourth function is the most complex, and that's where we see the activation of complement. Now, with complement, this is another protein that is used in the process of immunity. Notice the spelling, it's not with an I, so it's not, boy, your hair looks nice today kind of complement. Here, it's a protein. Now, the protein is not much bigger than an antibody. So once again, an antibody by itself and this protein complement by itself can't really do anything. But when they are put together, and that's done by binding the complement to the Fc region of the antibody, now it becomes a much larger protein, and it can be very dangerous. Now, the thing about complement, there are several different pathways, and they are quite complex. We're not going to focus on all of them. We're going to focus on one, what we refer to as the classic pathway, and that's going to be involved in what we refer to as cytolysis, which is the lysis of the cell. Now, the cell in this case is not one of our healthy cells. What we're talking about are gram-negative bacteria. What will happen now is that the individual proteins, complement, and looking at the antibodies, are small enough that they can move through the porins of a gram-negative cell. And when they do that and they move inside, now they will connect together. And once they connect together, they are big enough to lyse the cell membrane, cytolysis. And when they do that, they'll destroy the cell from within, but the cell wall remains intact. What this means now is that the wall with the endotoxin stays whole and is not released. Now, when it comes to all these functions, all the antibodies can do these except for IgE. IgE will only be involved in hypersensitivities, which we'll get to in chapter 18. IgD, remember, is only on the surface of B cells. So we're focusing on A, G, and M. Now, when it comes to these, your book always tells you IgM is the first to act, and then, well, that's not always true. It depends on where these antigens come into the body. The one thing I will mention, though, that's very important is when it comes to neutralizing toxins and the activation of complement, IgM is the most effective of all the antibodies at doing both of these. Number one, because it has 10 variable regions, so it can bind up to 10 toxins at once. And second, and that's why it's so good at neutralizing toxins. Second, it's found in the tissue already where the toxin is going to affect. When it comes to the activation of complement, it's the largest in size. So when you put this really big protein with another big protein, they become very good at lysing the cell membrane. Now, before we get into monoclonal antibodies, I need to talk to you about the secondary humoral response. I'm not going to give you a slide on this because it's a very basic thing, but I want to make sure you understand it before we move on. During the primary humoral response, understand that process is going to take several days to occur. It's going to take several days to get enough antibodies, and now those antibodies got to function in one of the four methods that we talked about in order for us to remove these pathogens. That takes at least a week. Well, the secondary response is going to involve those memory cells produced at the end of the primary humoral response. Those memory cells, once they're produced, are going to circulate around your body, either at the portals on the mucous membranes, in the tissue, or in the bloodstream itself. And what they're looking for is the reoccurrence of that specific antigen. So if that specific antigen re-enters the body, those memory cells have the ability to recognize it immediately. Once they recognize it, because they are plasma cells that just weren't functioning yet, they too can produce and release antibodies immediately. Those antibodies will then function in one of those ways we just mentioned immediately. So now what that means is this secondary response is immediate. The problem is you can't get to the secondary response without having the primary response. That's where vaccinations come in. Vaccinations represent the organism to your immune system. You undergo that primary response, and now you produce memory cells. These memory cells remain with you forever. Remember, they keep dividing and passing on that memory to future cells, and all they do is scout for that specific antigen to come in. And when it does, it produces antibodies, and they function immediately. So that antigen is removed before it can ever make you sick. The problem with this system, it only works on what we refer to as circulating antigens. If an antigen is circulating, it doesn't necessarily mean it's in the blood. What it means is it's out in the open. That means our immune system can see it. So as long as our immune system can see it, we'll respond with the primary humoral response and the secondary humoral response. But if it would get into the cells, then this system can no longer work. This system also cannot work when the antigen, what makes you sick, is actually something from you. What I'm referring to is cancer. A cancer cell is one of your cells that has lost its ability to differentiate and become a functioning tissue cell. Instead, because of mutation, all it does is keep growing. Now, as it grows and it just keeps multiplying, it forms tumors of these, or collections of these cells, tumors, and they will get bigger and bigger. And the way cancer kills you is in one of two ways. First of all, these tumors can just literally erupt right out of your body. They will literally explode right out of you. The second thing is that it will displace the healthy tissue. And now that tissue can't function. And if the tissue can't function, then the organ no longer functions. And if the organ doesn't function, the whole system shuts down. And if you lose any organ system other than your reproductive system, you die. Again, our humoral system cannot be used to fight cancer. Or can it? That's where monoclonal antibodies come in. Now, when we look at monoclonal antibodies, these are antibodies produced in vitro, meaning in the laboratory. We're going to take our knowledge of the immune system and the fact that all mammals have the exact same immune system and use that as a means to fight things like cancer. We're also going to see this used in the process of producing what's called biopharmaceuticals, which is something I talked to you about in previous chapters, and we will discuss here. Now, when we look at monoclonal antibodies, we can use any mammal. Here in this example, we're seeing a mouse. Okay? Now, with the mouse, we're going to note that it too has an immune system. And this immune system has B cells. Now, these B cells cannot identify the mouse as being foreign. So what we're going to do, though, is to allow it to identify what's foreign in us. Now, how do we do that? Well, the first step is we have to develop what's called a hybridoma. Now, that is a cell that is a combination of a cancer cell and a mouse B cell. Now, the cancer cell is not the cancer that's inside of the human. A cancer cell here, what we want to do is to give the qualities of a cancer cell to this B cell. Basically, cancer cells, because all they do is divide, become immortal. They never die off because they constantly reproduce. So if we can give this B cell that characteristic in laboratory, we can keep it alive. Now what we do then, once we create this hybridoma, we're going to mix it with a cancer cell from the human. Now, our immune system can't see this cancer cell as being foreign because it is one of our cells. The receptors are identical to all the receptors in us. So understand, when somebody has cancer, that means the immune system is not functioning like it should. So now what we're going to do is to get the mouse to identify it as being formed by mixing the cancer cell with the hybridoma. The B cell components will bind and become sensitized. When they do, they will differentiate into plasma cells, and the plasma cells will produce antibodies. Now, these antibodies are going to be collected, and then they have to be sterilized. Now, the way we do that is through filtration. Remember, you cannot use heat because that will denature this protein. If that protein is denatured, the shape is changed. Remember, these are locks and keys. If they are changed, then they will no longer function. And if they cannot, they will no longer bind to the antigen. And if they cannot bind to the antigen, then they're not functioning. So we've got to maintain them in their exact shape. The only way to sterilize things that are heat sensitive is to filter them, and that is extremely costly. Now, once we produce and release these antibodies and they're filtered, then they will be injected into the cancer patient. These antibodies will now bind up to the cancer cells. They won't bind to our normal cells because now they're able to identify things on that cell that our immune cells could not. And once they do that, the macrophages and the neutrophils will now see that these are cancer cells and will phagocytize them and remove them. Now, this sounds like a great way to treat cancer, and it is. The problem is cancer cells constantly mutate. Because they mutate, that means they will change the surface receptors. And when that occurs, these antibodies can no longer bind. That means you would have to go back in lab and recreate these antibodies. Now, guys, remember, that's going to take weeks at a time. And these cells are going to constantly keep dividing. So even though it showed promise for cancer, and it does work against some cancer cells that don't mutate as often. For example, if you have cancer of the kidneys, those cells tend to remain constant. This will work very well. But if it's something else that constantly changes, like what we see with certain skin cancers, this is not a great means of treatment. But would it be a good treatment if something remained more stable? Something like bacteria. Bacteria, although they do change occasionally, they don't change as often as what we see with things like viruses and with things like cancer cells. So what if we could go in and produce antibodies in mass to things like MRSA, a very specific form of Staphylococcus aureus? Or if we could produce antibodies specific to each of the 55 different forms of Streptococcus pyogenes that causes strep throat. We could then store these in mass, and then when you get that infection, put these antibodies right into your body. They would bind up to these antigens, these foreign cells, before they could ever get into your tissue. And now that would protect you against them. Now, folks, this would be a form of almost passive immunization, which we'll talk about later, because we're basically using antibodies as a means of treatment here and could be also as protection. But this is what biopharmaceuticals are all about. We would genetically engineer organisms to produce these specific proteins. We would then grow them in mass, sterilize them in mass, and freeze them, because freezing does not denature protein. And then when you need them, simply thaw them out and use them. The problem is the filtration process. It is extremely expensive. For one, let's say septicemia, you might need literally liters of antibodies. Well, one filter to do 500 mLs of these may cost several $100,000, and you can only use it 2 or 3 times. So it's just not cost effective. Once we learn a means of sterilization that is more cost effective, then we can see this being a process used. And the beauty is, look how specific it is. This would eliminate antibiotic resistance for 2 reasons. Number one, this isn't a chemical treating the cell. Instead, it's just a protein binding to it. It's identifying it to your immune system so it can be removed. It prevents the things that are produced from binding to tissues. It doesn't destroy them. So just by binding to it, if the organism would not recognize it as being something harmful and would not try to change. So this would be a huge advantage and would eliminate resistance. The problem is, it's probably going to be at least another 30 years before this will be useful. Now, monoclonal antibodies can also be engineered with poisons, and sometimes though this poison will be released into the cancer cell to kill it. So this is another thing we've tried. It's a little bit more effective, but the problem still is, in order to get the toxin into the cell, the antibody has to bind to it first. So as long as these cancer cells are constantly mutating, even engineering it with a toxin is not as helpful as we once thought. Now we're going to talk about the cell-mediated immunity. Now, this system is one we don't want to use unless we absolutely have to. It's the immunity system of last resort. So this immune system is only used with things that are no longer circulating. If those antigens are circulating, it's the humoral system that goes after them. But if they would get into the cells, or if the cell is a cancer cell, it's this immune system that responds. How does it respond? Well, it's going to use T cells. Now, T and B cells are lymphocytes. Now, lymphocytes you learned in Gen Bio produce protein. Well, the B cells produce antibodies. The T cells produce either enzymes or hormones. So that's where they differ. T cells do not produce antibodies, just hormones or enzymes. Now, there are several different T cells that we're going to talk about. The first type of T cell is what we call the cytotoxic T cells. We abbreviate them big T, little c. Cytotoxic T cells only work on viral infected cells. And when we say work on, they destroy that cell. Now that's a big problem because now we're seeing our immune system attacking us. Now that should scare you a little bit because folks, we don't want to destroy healthy cells. But the problem is this cell is infected by a virus. As we learned in chapter 13 with the lytic cycle, once that virus is inside the cell, the outcome is going to be death to that cell. So at this point, that cell is dead already. Destroying it is not going to be a negative. But what if the T cells now started destroying healthy cells? That's what we call autoimmunity. And that's why we don't like to use this system. It's right on the cusp of autoimmunity. Every time this system works, it's right on the cusp of going after healthy cells. So we really got to be careful with that. Now, when we destroy this healthy cell, that would be something negative. But here, it's already infected by a virus, so that is the positive. So you can see how close this is to being a negative. The other T cell we see involved is called a natural killer cell, the T NK cells. These destroy cancer cells. Every day, you produce about 5 cancer cells when your cells are undergoing division. Because every time your cells divide, the first thing they do is replicate DNA. Every time DNA replicates, at least 5 mutations occur. Now, those 5 mutations often are what we refer to as nonsense mutations, meaning they don't do anything. They're not positive, they're not negative. They simply occur in the junk DNA and they don't have any effect. But every now and then one gets through that is negative. And now it's up to this immune system to protect us. So if this immune system is functioning properly, that cancer cell is destroyed before it can ever produce a tumor. When people have cancer, that means this system is not functioning properly. In other words, they are immune compromised. Now, when we look at these natural killer cells and cytotoxic T cells, they have a problem in that they don't know how to identify these cells. Now that seems kind of bizarre. They're going to destroy the cells, but they don't know how to identify them. They are just like the macrophages. They are stupid in that way, and we want to keep them stupid so they don't destroy healthy cells. So once again, we're going to see that these cells, in order to identify what's foreign, need some help. The help comes from the T4 helper cell. Now, folks, the T4 helper cell is again going to produce and release lymphokine. But the lymphokine here does not cause B cell reproduction. Instead, it activates the killing T cells. What it does is it tells the cytotoxic cell, this is a viral infected cell. What it does is it tells the NK cell, this is a cancer cell. And it will cause the lymphokine, those cells, to produce and release something called perforin. Perforin is a hydrolytic enzyme. These cytotoxic T cells will then inject that into the viral infected cell. The NK cell will inject this perforin into the cancer cell, and it will perforate them. It will destroy them, and it will cause the cells to undergo lysis and destroy everything within it. So this is how we get rid of both. But once again, you can see this is right on the cusp of being autoimmunity. So we don't like to use this system unless we absolutely have to. Now, I have another question for you. My question is simply this. I know it doesn't make much sense when you think we're talking about cell-mediated immunity, but this question relates to all your immune systems. How does AIDS kill? Because I want you to think about this. Think about how many people died of AIDS last year. Okay? I want you to think about that, and then think about how AIDS leads to your death. We'll discuss this in class on Thursday. The last thing to talk about here is how the T4 helper cells are able to identify antigens. Well, the actual answer to this is we don't know. Your book tries to tell you, oh, this is how it's done. Well, those are just hypotheses. We are really not sure. As much as we know, there's still so much we don't know. And one of the things that we don't know is exactly how T4 cells can identify the antigens, but they do. And it's a necessity that they do so because it's needed for both the functioning of the humoral and the cell-mediated system. Now, the one thing we do see is that when they identify them, we know because they will now, once identified, release lymphokine. And that lymphokine, that hormone, will affect both the humoral system by speeding up B cell division and cell-mediated immunity by activating the killing T cells to function. But again, we don't really know how they're able to identify these things. Now we're going to move into chapter 17 on immunizations. Now, the first immunization we're going to talk about is one that is active. Now, an active immunization means that your own immune system is stimulated. That means you're going to get memory. The problem is it's extremely slow to do so. So the benefit is memory, but it's going to be slow to gain it. Now, there are two types of active immunizations. The first type we're going to talk about is a vaccine. Now, vaccines are where we inject the organism or parts of the organism into you to stimulate your humoral immune system. Now, when we look at vaccines, there are two types. What we call dead or inactive or attenuated. Now, if it is dead or inactive, it means that it has no ability to affect you. If it's attenuated, remember what that meant. We learned this in chapter 14. It means we have reduced its virulence. The organism will still produce disease, but if it does, it won't be as bad. Now, to understand these two, we're going to talk about, for the dead or inactive, the Salk vaccine for polio. The attenuated vaccine, we're going to talk about the Sabin vaccine for polio. Now, the Salk vaccine was one that was developed first. It was made from the inactive virus, and it was put into a needle and injected into you. The Sabin vaccine is the attenuated form. The virus is active, but it's been weakened and done so by transposing the virulence, growing it in a different life form. And then this one you take orally. So you would, it was put into like a sugar cube, and you simply put it on your tongue and swallowed it. Now, when we look at vaccines, their effectiveness is going to depend on some things. First of all is the type. Now, when we look at the Salk vaccine, it is not as good at producing immunity as the Sabin. And the reason is because it uses inactive or dead. When something is dead or it's inactive, that means we've used something to kill it or inactivate it, which changes its shape. And because the shape is not perfectly the same when it's dead or inactive as it was when it was virulent, alive, that's a problem because our immune system, remember, is a lock and key. So the vaccine, when it is inactive, the problem with that is it doesn't always give you the best type of immunity. Now, the second one, the attenuated one, will give you wonderful immunity because it's the real thing. The problem is it can also make you sick. So keep that in mind. When we look at vaccines, remember too that these can only be given to people that have a fully functioning immune system. Remember that, because if you put an attenuated vaccine into somebody, that would be a big problem if they are immune compromised, because they would get this disease, and in them it may not be mild, it may be serious. One other thing to talk about here, remember I said injections of parts. When you get a flu shot, you are not getting the organism. It's just the proteins on the envelope. That's why it's impossible to get the flu from the shot, because you're not getting the organism. You're just getting the proteins. Now, the second thing the vaccines will depend on is the storage. Remember, these are all protein. And because of that, if you store them improperly, such as in a hot warehouse, they will denature. And so when they're injected into you, your immune system will respond and produce antibodies, but they will be to the wrong shaped antigen. And so now when the real thing comes in, you'll have no protection. The third thing that will affect vaccines is the route of transmission. And this again is why the Sabin was more effective. When it comes to polio, the way you get it is through a vehicle transmission by ingesting it. You don't have it go through your skin. It doesn't go through your eyes. You swallow it. That's why a lot of people got the polio from swimming in water with other people that had the disease. They would release it into the water and people would drink it, because yourself, swimming in a pool or even in a lake, you will sometimes ingest a little bit of that water. Now this organism would go in. Now, when we use a vaccine to follow that route of transmission, we get much better protection because the protection is right where the organism will be found. Now, these vaccines only work when we're dealing with diseases produced by direct action or viruses where the virus will be present, or if it's a eukaryotic parasite, when the parasite is present. But if the disease is due to a toxin, these won't work. What you use instead is a toxoid. Now, this is a protein that resembles a toxin. Oid means to resemble, toxo means poison. A good example of this is the DPT vaccine. Now, this is a protein made from pertussis. Pertussis is whooping cough, and that disease is caused by a toxin. Now, in these three, diphtheria, pertussis, and tetanus, pertussis is the weakest of the three, and all three are caused by toxins that look very much alike. So what we did was remove that pertussis poison and tweak it just a bit so it no longer can make you as sick, hopefully not sick at all, but not as sick. And now, since it resembles these other two, which are life-threatening, when we put it into you, now your immune system can produce antibodies to neutralize these toxins before they can affect you. Now, the only problem is because this isn't the real toxin, our immune system sometimes tends to forget what it looks like. So we have to remind the system by doing booster shots. That's why you have to get your DT vaccine boosted every 10 years. Now we're going to talk about passive immunizations. Passive immunizations are when we create immunity in another organism. Now, when we do this, the benefit is it's extremely fast. We don't need to undergo primary humoral response because we're not using our system. We're using the immunity created in another life form. Now, the downside of this, it's not going to be long-term. It's very short-lived. So with this type of immunization, it's going to have to be repeated if it's going to be protective. Now, some examples of this or types. The first type is when we use gamma globulin, and this is something we commonly use when our soldiers go off to war. This is where we are going to put into them antibodies produced by another life form and introduce it into their bloodstream. Now, the way we do this is we're going to go out with horses, cows, and pigs and inject them with the etiological agents of the diseases that our soldiers might encounter in a foreign land. So when our soldiers were going off to Afghanistan, they were injected with this gamma globulin. Now, when the etiological agents are put into horses, cows, and sometimes pigs, remember, the immune systems are the same as ours because we're all mammals. Now, these etiological agents may kill those animals, but before it does, their immune system will start to produce protein. Since they were injected, those antibodies will be IgG produced in the bloodstream. What we do now is to remove the blood of those animals. We spin it in a centrifuge to separate the blood, the cells, from the plasma. And now in the plasma, we are going to run that through a filter and remove the antibodies. Those are put into a saline solution, and now we inject them into our soldiers. Now that means for about 3 months, they're going to be protected against anything they may encounter in that foreign land. The problem is, if they're there for longer than 3 months, they'll have to be re-immunized with the gamma globulin to give them further protection. This is also the same kind of process that we see naturally occurring when we see mother to offspring and a child breastfeeds. It's a very similar process. Now, the other means is antitoxins. Now, gamma globulin was more preventative. Antitoxins are not preventative, they're used for treatment. And a good example of this is when we look at the immunoglobulins produced in another life form that we will use to neutralize toxins that get into us. And a good example of that is when we're talking about snake bite. Now, when you're bitten by a snake, that's a poison that's put into you. The problem with an antitoxin, it's very specific. Remember, locks and keys. So we have to know exactly what snake bit you. You can't say I was bitten by a rattlesnake. Was it an eastern diamondback? Was it a western diamondback? What is a Massasauga rattlesnake? Was it a timber rattlesnake? Was it a pygmy rattlesnake? Was it a sidewinder? There are so many different forms, and you have to know exactly which one because they're locks and keys. Now, the way we set up antitoxin to get it ready for you is that we milk snakes. We take their venom and we introduce that venom into other mammals. Now, again, that may kill the mammal, but before it does, they'll produce antibodies. Those antibodies are going to be removed again by plasmapheresis, where we separate the blood cells from the serum, and then in the serum we will filter that to remove the antibodies. Now, those antibodies will be stored by freezing them, because remember, proteins are not denatured by cold. And now when you need it, it'll be thawed out and administered. The problem is an antitoxin has to be administered right away because it has to be given to you before the toxin has a chance to bind to the target tissue. That's why the longer it goes when you're bitten by a snake, the less likely that this antitoxin will help you. The same applies when antitoxins are used to treat certain diseases that we'll talk about later. The longer we wait to administer the antitoxin, the less effective it is. Now, with all immunizations, whether they're active or passive, we have to worry about side effects. And when it comes to side effects, the major one is allergic reaction. Now, we're going to talk about hypersensitivities, allergic reactions, in Chapter 18. But understand that anything, and I mean anything, could be responsible for an allergic reaction, including an immunization. The other thing we have to worry about is when we're using an attenuated vaccine, and that is when it produces disease. Remember what I said earlier. Someone that is immune compromised cannot be given a vaccine because there is always a chance it could induce disease. Now, some doctors will say they can still be given dead or inactive vaccines, but there are others that even refuse to give those. But if it's attenuated, that's a serious problem. It's also a problem in a pregnant woman because although it won't hurt the mother, it could cross the placenta and induce the disease in the developing fetus, which could prove to be fatal. Now, this is the material in chapters 15, 16, and 17. Go through this, take any notes you need, and please write down any questions you might have so we can go over those when we meet in our next class meeting, which will be in Thursday's class.